Okay, while you watch this video, you should take some notes on, well, first of all, who Gregor Mendel was, and then what we're going to get into is using this basic heredity that you guys have learned about. Now we're going to be able to use that to solve genetics problems and figure out probabilities that a certain set of parents will have offspring with particular traits. And we're also going to start getting into some types of genetic crosses that are not exactly as Gregor Mendel would have predicted, but that's a different set of notes. This one is just going to be about how do we use this to solve genetics problems. All right, so there was a scientist, he was actually also a monk, that was Austrian. He lived in the 1800s. He did most of his work in the 1850s and the 1860s. And he figured out all of these rules of heredity, that each individual has 2 alleles for each trait, and they're located on different chromosomes. Well, he didn't know about chromosomes, but that's what we now know is the case, that they are located on different chromosomes, and that each parent can only pass on one of their 2 traits to the offspring. He also figured out how dominance and recessiveness works. And so Gregor Mendel was really pretty amazing that he figured this out because he couldn't even see it. We didn't have the right technology to see chromosomes with microscopes yet. So he did this without being able to see that the chromosomes have homologous partners and that the homologous partners separate from each other. Now that we were able to see that, it just really confirms what Gregor Mendel figured out, kind of in what we would describe as a black box. He did it using probabilities. So what Gregor Mendel always did is he always did crosses where he started out with individuals that had the opposite phenotype. Okay, so this would be his first cross. He called it his parental cross. And for this parental cross, he always started out with individuals that had 2 alleles that were the same. Okay, we call that homozygous. That's coming up in one of the slides. So in this case, he started out with one plant that had 2 recessive alleles and the other plant that had 2 dominant alleles. Mendel called this true breeding. And then what he would end up with in his next generation here is that because this parent can only give a little t and this parent can only give a big T, all of the offspring from that cross would have 1 big T from that parent and 1 little t from that parent. That happens in meiosis. So then the offspring would have 1 dominant allele, 1 recessive allele, and they would be all tall. Okay, so that would be what he would see in the first cross that he did. But he didn't stop there. Gregor Mendel went on and did a second cross. Okay, so this is the cross we talked about already. And in the second cross then, he would look at those individuals from the first cross. Okay, remember these guys from the first cross, they're big T, little t, and he'd allow them to self-fertilize. And pea plants, lots of other plants will do that where they use their own pollen to fertilize their own eggs. Okay, so the cross is self, so big T, little t, cross with big T, little t. And what he ended up seeing every time he did this is that the generation after that, so these are the grandchildren of his parental generation, this generation here always had a ratio of 3 dominant individuals to 1 short individual. So 75% of the offspring would show the dominant trait and 25% of the offspring would show the recessive trait. So Gregor Mendel figured out using this information what dominant and recessive was, and that these individuals then can only pass on 1 of their 2 traits, and based upon the possible combinations, you would end up with 3 individuals showing the dominant trait and 1 showing the recessive trait. Okay. So Mendel repeated this. He did this, it took him many years to do these experiments. He repeated this not only with tall plants and short plants, but he repeated it with purple flowered plants and white flowered plants, green seeds and yellow seeds. So there's 7 other traits, and he always ended up with, in the grandchildren generation, so after his second cross, a 3-to-1 ratio, dominant to recessive. All right, so in thinking about that and how that works and why that works, we do need to know a little bit about, there's another set of terms, one more set of terms that we need to go through here in order to work with these genetics problems. I've mentioned this to you guys already because it was in that video with the peas. Homozygous versus heterozygous. So if we have here, we're doing Rudolph the Red-Nosed Reindeer and his family, if we have an individual that has a black nose, okay, so let's say that the black nose trait is a dominant trait, then that means that that individual can either be big N or big N little n. Now I've given to you in this particular problem here that this individual here is big N, so he's showing the dominant black nose, and the characteristic or the homozygous versus heterozygous term that relates to that would be homozygous. Homo means same. So because we have two of the same alleles here, that's homozygous. Now you can also have homozygous for the recessive allele. So if we take a look over here like Rudolph, okay, little n, that's two of the same. Both of those alleles are recessive alleles, and so you end up with the recessive trait and the red nose. However, we still do have this described then as being homozygous. So usually when you're talking about homozygous, you have to also qualify it. The first one here, this one is homozygous dominant, whereas this individual is homozygous recessive for their genotype. Of course, then, the opposite would mean that the individual has one dominant allele and one recessive allele. One dominant allele, one recessive allele, like this individual in the middle. Of course, they're showing the dominant trait then, and then what we would describe as their genotype then would be heterozygous. Hetero means different. Okay, so keep that in mind as we're doing genetics problems so that you can pick out from the information how to solve it. Okay, so now let's go into some thoughts with this. So thinking about both of Rudolph's parents, they did have black noses. What was Rudolph's genotype? You pause and think about that. Okay, Rudolph had a red nose. So Rudolph's genotype would be homozygous recessive. I could use N's. I don't know, I picked H's there, but I could use N's. So knowing that Rudolph is homozygous recessive, okay, there I used little r, we should be able to figure out what Rudolph's parents must have been. So Rudolph's parents both did not have the red nose. They had the black nose. So they must have had a dominant allele to have a black nose, but because they both were able to give a recessive allele to their child, Rudolph, they must also have been a carrier for that recessive allele, and their genotypes were each heterozygous. Okay, so be thinking about things like that. Okay. So let's take a look at some problems that are very typical to what you're going to be doing. So these are like word problems in math class. You need to read through them and you need to pull out of from there the information that is the most important and then go through and solve the problem. So the first thing that I always do when I'm dealing with a genetics problem, even now, even after doing this for so many years, I look at the problem and if I can, I pull out and write down the genotypes of the parents. So in this problem it says, if two parents are heterozygous for eye color, what is the chance that their child will have blue eyes, which is a recessive trait? By the way, that's very much of an oversimplification, but we can work on it. Okay, so both parents are heterozygous for eye color. We can write that down. Heterozygous, and then here's the cross. Okay, so now we've got both parents. They're heterozygous for eye color. Okay, the next step in solving a genetics problem is to make a Punnett square. Okay, so you probably have done these Punnett squares before in 7th grade, but we certainly will go through a quick refresher here. A Punnett square, all it does is it's a tool that'll tell you possible combinations that you can get from a single set of parents. And what you're going to do is you're going to keep in mind that each parent can only give one of their two alleles. So what you do is you take one parent's alleles and you separate them down the side. Okay, this parent can give a big B or it can give a little b. And then the other parent's alleles you also separate and you put them across the top. Big B, little b in this case. Then what you're going to do is you're going to pair together the letters into the boxes, showing the possible combinations that then an offspring could have. So this parent could have, or this child could have, Big B, and they would have brown eyes. They can have Big B from there and Little b from here. Okay, so you see we're following and putting them together in the boxes. They could have Big B, or they could have Little b. Okay, so follow from the boxes where they would go all the way over into those boxes. All right, so the question is, then the last step in solving a problem is answer the question. So in this question here, it's asking for what is the chance that their child will have blue eyes. Okay, so in this box, if you take a look at the Punnett square, this is the only condition here where the child has blue eyes. All of the others are showing the dominant brown-eyed trait. So there's 4 boxes there, and only one of them has the condition we're looking for. So it's 1/4 chance. The other way you could express that and still have the same answer and have it equally correct would be to write it as a percentage. So it's a 25% chance that you would end up with that particular combination. Okay, now I would like you to pause this video and see if you can solve the genetics problem that's indicated on the slide. Okay, so notice here now in this problem I have written down the genotypes of the parents. The woman has attached earlobes, which would be the recessive condition, and the man has unattached earlobes, but the problem said that he's heterozygous because his father did not. And so we make a Punnett square next. Okay, so there I have the Punnett square constructed and completed. And then the last thing you need to make sure is that you answer the question. And so in this particular case, the question's a little different than the last question. It's how many of their children would you expect to have unattached earlobes? In the previous section, it said that they do have 6 children. All right, so looking at this, we want to know how many would have unattached earlobes, which is a dominant condition. So we've got unattached earlobes in both of these individuals here. So you would expect half of their children to have attached and half to have unattached earlobes. So half of the 6 is 3 children. And there's your answer, 3 children. All right, now that you've completed the notes on this section, there is a Punnett Square practice worksheet where you can try a bunch of these different kinds of problems, review some of the vocabulary and terminology for many different traits in humans. So that would be the next thing for you to do. Thank you.